Hello and welcome to Lab Down Under. Today I'll be talking to Shannon McCluskey, who did some research on dolphins here at Murdoch University in Western Australia and who is now currently residing in Washington State. Her research looked at the eating habits of dolphins off of Western Australia, basically breaking populations up into two parts and examining what their dietary habits were. Shannon talked to me about her research, what it was like to fish through a dolphin's stomach, and how it fit in to all the conservation efforts that we need to bring to save this beautiful species. I hope you enjoy the interview. Yeah, first, I guess, thanks for taking the time out to sit down with me on the Lab Down Under. This is good. I started this video interview thing as well to kind of expand the blog, so thanks for that. I wanted to talk about your research on dolphin foraging habits. So basically, you looked at a specific type of fish and the diets that these dolphins around Australia had. So first of all, broadly, how does this kind of help our wider conservation efforts?
Who cares?
Yes, basically, yeah. Got to have a hook to get people in, right?
Bother. Well, I think at the most base level, if you don't have adequate nutrition, if you don't have enough to eat, then nothing else matters. Mm-hmm. You don't have the fuel to grow, to reproduce, to have a functioning immune system, to move around, to perform various behaviors. So it's a really basic biological need to have not just food, but the right kinds of food, so quality as well as quantity. And so as from a management perspective, you don't know what that means or what your priority is for managing, in this case, fisheries, recreational, commercial fisheries, or, land development, contaminant loads, anything that might affect those prey species. You can prioritize where your money and your effort goes. Also, on a big picture level, it's really important to understand, the roles predators have in systems. They have a huge impact on the ecosystems that they inhabit, and without knowing the prey species that they're relying on and the effects that those prey species have, we really don't have a very good understanding of the system. It wasn't until we understood how important sea otter predation was on sea urchins to maintaining kelp forests. And then, of course, there's just a multitude of organisms that rely on a healthy kelp forest. But without understanding, that foraging dynamic, we didn't protect sea otters for that reason, or likewise wolves. In reintroduction, Yellowstone really highlighted the fact that predation on ungulates like deer and moose and elk were absolutely critical to the health of the riparian ecosystem and the reforestation of areas that had been suppressed from growth because of browsing. And we would never have, guessed that the freshwater fish in those rivers were absolutely dependent on wolves. Bottlenose dolphins, which I've studied, or killer whales, which I've studied, are predators in the systems that they live in, and they have these kind of keystone effects as well. So it's important to understand what those linkages are, so that we can protect those various links. We can also know how maybe contaminants might flow through a system if we know who's eating who.
So your study, you looked at dolphins. First off, I just wanted to get this right off the bat. The study that you looked at, the dolphins that you looked at in the study, these were all ones that died of natural causes, right? These were ones that you've kind of found on beaches and things like that?
The dolphins got stomachs from were all they all died and were opportunistically collected.
Yes.
If we could get to them before they turned into complete mush. The stable isotopes samples from dolphins came from live animals, and that's just a little dart that takes a tiny little bit of skin off. So they weren't the same individuals. So isotopes were from live animals, the stomachs were from dead animals.
Okay, so can you explain a little bit, because that leads into the next question that I wanted to ask about looking through the stomachs and then the stable isotopes. So can you explain a little bit more about how those two methods kind of worked, and then how they work together?
Yeah, sure. So stomach content analysis is literally looking at the hard parts that are left over in a stomach that you can recover. So the benefit of that is seeing what's actually ingested. I mean, without a doubt, that got into the stomach of the animal when you're looking at, well, a lot of animals, but specifically ones that spend their life underwater and don't necessarily come to the surface to show you what they're eating. It's really hard to know what they're actually ingesting. And just because you see animals, milling at the surface or appear to be chasing something, you don't know if they've caught it, you don't know if they've consumed it. So looking at stomach contents is a way to look at exactly what has been ingested, and then you can compare what relative frequency different prey species have been consumed at. And of course, it's very specific. I mean, you can get down to species specificity. Not always. It depends on the condition of what's left behind. You're happy to get an identification down to family level, but it can be very specific. And then you not only have species, but depending on the species, you can potentially get age of that individual, relative length of that individual, if there's been prior studies on that. So that's all really, really important information to know, like what age classes of, say, fish are they consuming? But it's a short time frame. It's limited to what this animal has ingested in the last hours, maybe days, at the most weeks. So the time scale is really short. Whereas stable isotopes is a much broader time scale, and it depends on what tissue you're looking at, what the turnover rates are. Those time scales are, weeks to months or even years if you're looking at bones. So the time scales are quite different, but stable isotopes is giving you a much broader and longer, kind of like the stepping back big picture view, and the stomach contents are giving you the snapshot in the moment view that's much more specific. And so together they're like using two pieces of the puzzle to try to sort out the picture instead of just one.
What were the key findings then of the study, and was there anything that kind of popped out and like really surprised you?
Well, we were comparing what we call inshore dolphins, there's a group of dolphins in this area that hang out all year round, and it's mostly mothers and their dependent offspring. And then there's a group of dolphins that come in the summer months. And so the population in this area is like 180 some animals in the summer months and like less than 75 in the winter. So there's this seasonal fluctuation. And one of the original questions I had was, well, why? What's driving these movement patterns? And are there differences? Are there foraging differences between these two groups? And it turns out that there are foraging differences, that the isotopic signatures say that they're foraging on different things anyway, which means that they're two groups of dolphins that come together. They share habitat. They intermingle. They certainly breed together, but they're occupying different ecological niches. Mm-hmm. And for that reason, they would probably take some different management strategies. And you can't just necessarily lump a group of animals together just because you see them together. You know, just like the killer whales behind me, they have sympatric ecotypes, which might share territory or have overlapping spatial distribution, but they have, you know, very different life histories, and they have very different needs and should be considered, you know, kind of separate populations.
Okay.
And they were, we saw specialization in their diet. They were not just eating things because they were there. They were not eating things in proportion to what was available in the environment. So there's some specialization, which again is a reflection too on quality of prey. They're really selectively foraging on prey that is more calorically beneficial, you know, caloric value, more bang for your buck.
Yep. Okay.
So that again, you know, helps us prioritize what species to manage well. I think one of the most surprising things for me was how small some of the prey items were. Some of these fish, actually a lot of the fish that I was identifying in the stomachs were little gobies. They're like this big. And you have to eat.
You said bang for your buck though, but must eat a lot of those things.
You have to eat a lot. I mean, calorically, they're pretty high value, but you still have to eat a lot. But it was juvenile animals that were eating these smaller prey, and I expect as they grow to be adults that they're able to catch bigger, faster prey.
Right.
Instead of snacking constantly on these little bite-sized morsels.
I can identify with that, the constant snacking though. So like, oh my God. So probably something a little bit tastier than a goby. Okay. Do you know why the dolphins had split their diet and their predation habits in this way, or is it kind of too early to kind of say that?
Usually when you see this niche partitioning, it's a way to decrease competition between groups of animals. You're like, well, I'll specialize in eating this and you specialize in eating that, and we can still share the same habitat and not directly competing with each other. Dolphins, like other social predators, are relatively long-lived. You know, they have a relatively long juvenile period, and their foraging strategies are learned from their mom. So we do see some really specialized foraging strategies in bottlenose, like up in Shark Bay with the sponge feeding and so forth, and using conch shells to trap fish. And those are all learned behaviors that you can trace through matrolines. And so a big part of it is probably just that this is what I learned from my mom. And this learned how to catch and how to catch it. And it took me years to perfect this. So this is what I'm going to do.
It works. So why do anything else, right? Yeah. Okay. Interesting. So I was going to ask, with the dolphins that you looked at were just off the WA coast. Now, can your findings then, are they specific to that population of dolphins, or do you think you can extend them to kind of dolphins elsewhere around the world?
I don't think you can necessarily say, well, this is what these dolphins seem to eat, and so therefore that's what other dolphins are going to eat.
Mm-hmm.
I think what you can say is that this is another example of dolphins that are more specialized in their foraging than what we previously thought. It was considered, they were considered more generalist predators than they really seem to be.
Mm-hmm.
But there are some overlaps of, at least on the family level, of fish that are important for these guys. And I think the fact that they are eating smaller fish is something that should be applied to other systems, to help us, again, prioritize our conservation efforts, and to accentuate the fact that it's not maybe just the biggest, flashiest fish that the dolphins are going for and that we need to conserve, but it's these small, easy to catch species as well. And I think it also reiterates the point that because of these specializations and this long time frame to learn these specializations, that animals like dolphins can't adapt too quickly to huge changes in their environment. So, you take away their prey resource, you change the environment through, climate change and overfishing, or, massive contamination. They're not necessarily going to be able to just switch prey.
Mm-hmm. Yeah. It seems like there's a lot of pressures on dolphins. Just in general. You mentioned climate change, and you mentioned overfishing and pollutants and stuff like that. It's just a lot more needs to be done, I think, to protect them, which is what this
For sure.
Finally, then, if my readers wanted to kind of follow you online to find out more about your research, where can they do that? Are you active on social media? Do you have a university webpage?
I am developing. It's in its infancy, really, but I do have a Facebook page that I am posting some research related information on, which is under thebluemuse.org, and Instagram of the Blue Muse as well. There is a university lab page, but it's a bit outdated. Aquatic megafauna research unit at Murdoch University.
Thanks very much, Shannon, for taking the time out for this. It's been good.
My pleasure. Thanks for reaching out.
Well, I hope you enjoyed that interview. If you want more information about Shannon's research, I will leave a link to the lab down on the blog and her social media pages below. Also, I'll leave a link to my Patreon, where you can come support the blog and hopefully help it expand and grow in the future. There will also be an extended interview for my Patreons on the Patreon page, so be sure to check that out. Well, until next time, keep on being curious, and I'll catch you around. Bye.